Put like rails in there, then also we can set some card in there.
Okay, it's 6 o'clock. We'll be starting in just a minute, and we look forward to this program. I want to give people a chance to get here because it's right about 6 o'clock. Okay, so I wanted to introduce myself. I'm Jean Wilson. I am an officer of the Redbud chapter of the California Native Plant Society. I am pleased to be able to welcome you to this program on behalf of the Redbud chapter. And we're looking forward to hearing about research on the North American redbud species. And Chrissy will tell you a little bit more about that in just a minute. The Redbud chapter was founded about 30 years ago in Rockland. And we've grown over those years to over 400 members. We offer a wide variety of services and activities, and including things like advocacy, conservation, horticulture, education, science, field trips, and programs such as this, as well as twice a year plant sales in Nevada and Placer County. We have published 2 comprehensive guidebooks to the native plants of Placer and Nevada counties, featuring 800 wildflower, tree, and shrub species. And those are available in local bookstores and online from the CNPS store. I wanted to let you know that our fall plant sale is coming up sooner than it'll be here pretty soon, let's put it that way, on October 1st. And we are going to have an online sale again, because that's the most effective way to manage it. And there will be a members only sale from 8 a.m. to 1 p.m. And then starting at 1 p.m., everyone will be able to buy. And we welcome you to that. And there are more information. If you'd like more information about the sale and other activities of Redbud, you can sign up for our newsletter. You don't have to be a member. Just send an email saying that you're interested and you would like to have us mail you a copy of our newsletter when it comes out about 6 times a year. And that email should be sent to, and I'll put this in the chat in just a moment, but it should be sent to Native Plant Help, all one word, no caps, no punctuation, Native Plant Help at redbud-cnps.org. Give your name and say that and your email address you want the newsletter to be sent to, and a request that we add you to our mailing list so that you will receive the newsletter. And at that point, and Chrissy, you can take it away.
Okay. Okay, thanks very much, Jean. We are really looking forward to learning more about the evolutionary and adaptive history of our namesake plant, the redbud, here in North America, including, of course, our redbud, the Cercis occidentalis. And here to do this is Camille Noel. I first met Camille at the Redbud nursery when she had come to volunteer, and I had also. And when I learned that she had done her master's thesis based on studies she had done on the redbud, the light bulbs just went on. It was like, you have to share this with our redbud community. And I could not be more pleased that she is here this evening to do that. And I'm very pleased that you all are here to share in this. So for those of you who haven't filled up on the background for this, just to let you know that Camille earned her MS from San Francisco State in partnership with the California Academy of Sciences, which is a great place, the one in Golden Gate Park, the public part of it. Her MS is in ecology, evolution, and conservation biology, with an emphasis on California native flora. Is this perfect or what? So her research on Cercis, during that time, she went throughout the parts of California where the redbud grows natively and the Colorado Plateau. She has also worked on projects involving several of our larger bird species in the East Bay, the barn owl, the golden eagle, and the prairie falcon. And she has come now to live in Nevada City as of 2021. And she is, of course, a member of the Redbud chapter. How perfect it must be that she can say to her friends, see, I now belong to a chapter of CNPS that was named after my master's thesis. Anyway, so with that, I would also like to let you know that yes, you are muted, and yes, we do not see your pictures. You won't see mine in a second. We're just going to have our speakers video be showing during the presentation, and you can ask questions at any time during the chat, and I'll gather those up, and then we'll ask those of Camille when she's done with her presentation. If during that time you see a question come into the chat and you go, oh, I know about that, feel free to join in the conversation that's over there in the chat. And so with all of that, then I will turn this over to Camille. Camille, we're so glad to have you here. Thank you so much.
Thank you, Chrissy and Jean. What a pleasure to be here. I really appreciate that enthusiastic welcome, and it is wonderful to be in the Redbud chapter. Welcome to all of our guests here. I hope to meet many of you in the future in person and see your faces. I hope you're all doing well. I hope you're comfortable and maybe grab something to drink and munch on. That's gonna help me learn new things when you're comfortable. I appreciate you joining me for this talk. Thanks to the CNPS Redbud chapter for facilitating and promoting these talks. My presentation is titled North American Cercis, or Redbud, a study of evolution and adaptation. Here's a picture of the California Academy of Sciences. I hope many or all of you have been there. This is where I conducted my graduate research. It's a historic and remarkable institution. It's the oldest scientific institution west of the Mississippi River. It was founded in 1853, so it predates all of the Western universities. It was and continues to be a central hub of biodiversity science on the West Coast and is one of San Francisco's most popular tourist attractions. One of the main questions investigated at Cal Academy is, what is a species? How many are there? What defines them? How do we know? So that's something I'd like you to keep in the back of your mind. Here's my title slide from my thesis defense, North American Cercis, a morphometric and phylogenetic study. My advisor was Dr. Peter Fritsch, who you'll hear a lot about. And on my thesis committee were Drs. Tom Parker and Jerry Davis from San Francisco State University. Like we've said, I was fortunate to collaborate between these two institutions, which was really incredible opportunity. The genus Cercis includes all of the species commonly known as redbuds. And I'm going to talk about the genus Cercis, the historical recognition and uses of Cercis, some characteristics and the distribution, previous taxonomic studies and the authors of those studies. And I'm going to talk about my morphological, adaptive trait, and phylogenetic study. What I'm not going to talk about is how to grow your own redbud and why your own redbud is happy or sad. I do get a lot of those questions, and It's not my specialty. I'm sure there's many other people that can help with that. I also was asked when I started on this project, and still people ask me, like, why are you so interested in Cercis or the redbuds? And so before I go much further, let me explain. It really started with Dr. Peter Fritsch. I was accepted into his lab at California Academy of Sciences in 2011. And he was a curator in the botany department. This is him in the Yunnan province of China. And Cercis was of interest to him. He proposed a number of different plant species and projects. And after looking at them carefully, I decided that Cercis was it for me. Also wanted to study a California native. People do work from around the world, but it was clear to me that I wanted something that I could go out throughout our incredible state and our diverse flora and see in the wild. Another thing I found fascinating about Cercis is due to its disjunct distribution, which I'll talk about more because it's in China and in the Mediterranean and also throughout North America. Research in this group can be used to test models of historical biogeography. In other words, Cercis phylogenetics can be used to inform theories about the distribution of plants and animals during earlier periods on Earth under different climatic conditions. Cercis is in the bean family, the Fabaceae. It has a typical legume type of fruit. This is Cercis in the middle, Cercis fruits, and these are more typical peas on the side. It's a flat pod that contains several seeds. The other pictures are cultivated green beans. The legume is a trait shared in the bean family and a primary form of evidence for the common origin of all the species in the family. Many beans fix nitrogen through nodules on their roots, but nitrogen fixation has not been found in Cercis or closely related taxa. This is a phylogeny of the Fabaceae group, the family. This tree shows an origin of the Fabaceae at 65 million years ago, right at the end of the Cretaceous period. The tribe Cercidae that I'm talking about occupies a basal position that diverged from the rest of the group around 34 million years ago from the inference methods we've used. Other common and noteworthy groups are the Faboideae, including our garden peas, the Mimosoideae, including acacias right here, and the Caesalpinioideae, which I have here, which includes the Senna armata, which is a beautiful plant from the Southwest. There's Cercis showing there in the basal position. So focusing on Cercis now within the bean family, commonly known as redbuds. There's 10 species throughout the world. There's a little debate about how many are in China, but we can round to 10 species. They're winter deciduous. They lose their leaves completely. They're woody. They grow up as multi-trunk shrubs, quite large to trees, and the flowers are showy and rose-colored. They're easy to recognize, generally. This is a western redbud from Northern California. It grows as a multi-trunk shrub to about 12, sometimes even 15 feet. The alternate arrangement of leaves on the branches hang in this wonderful regular pattern that at times reminds me of a decorated Christmas tree. Cercis has simple leaves. The leaves on the western redbud are semi-rounded to kidney bean shaped. The ends of the petiole also has a pulvinus, like a mimosa does. So the leaf can move due to contractions in the cells here. You may be familiar with this from prayer plants as well. Cercis is cauliferous. In other words, the flowers grow directly from the branches and main trunks of the tree. Their flowers are adapted for pollination by insects, including many types of bees. The flowers have a typical pea or bean form with 5 free petals. Here we go, 5 free petals, which are divided into 2 wing petals. These are known as the wings, a central banner petal, which is in the front of the wings. And then these two hook together to make a keel. Inside, this is useful for identifying the subfamily. They have a short cup-shaped hypanthium, and inside the keel are 10 stamens and one elongated superior ovary with a curved style. In the subfamily, the flowers are bilaterally symmetrical, so you can divide it in half and get an approximately mirror image. The plants produce prodigious numbers of fruits throughout the year from my observations. You can see tons of fruits on this plant, and they become browner, which I'll show you in a moment. When it really dries, it's brown, and then you get this wing, which was something that we measured, and it kind of shows the evolution of more recent species in this group. The dry fruit is formed from a single carpel and contains just a few seeds. When the pods mature, they turn a dark red to brown color. And I've seen plants that seem to hold them for 2 years. They don't necessarily blow away right away. So where does this genus occur? It's found on 2 continents, but it's restricted to the northern hemisphere and mid-latitude warm temperate climatic zones. The genus is thought to have originated in China, like I said, around 35 million years ago. So this is the earliest group. And then there's another one here, it's called the Afghan redbud. And then there's another species here in the Mediterranean, Cercis siliquastrum. So this is 8 species. And then what we have in North America is considered 2 species currently. This is a better picture illustrating where they are in North America. It's a large and fragmented distribution, which is shown in green. And it includes a number of remote isolated populations. The solid green includes the wet adapted form, and the dotted green indicates the dry adapted form. The central and eastern section contains plants that are adapted to both wet conditions in the north. So this is wet, dry conditions in here throughout Texas and northern Mexico, and then wet again in the south. This is in the Sierra Madre Oriental Mountains of central Mexico. The western section of this group is dry adapted and works well with summer drought. Some habitat photos. This is a mesic or wet habitat. Cercis occurs in the understory in US hardwood forests, and pictures here of the Sierra Madre Oriental in Mexico. And then xeric habitats. So this is a typical California forest with gray pine in the back and very dry and exposed foothill woodland. This is from Snow Canyon in Utah. I'm sure there's flash flooding through here, and Cercis can hang on into rocks and thrive in these dry canyons. So now I want to talk a bit about previous uses and scientific study of Cercis. Here I list the most noteworthy groups and individuals that I've come across in the many years I was looking at this that have used or studied Cercis. In the next slides, I'm going to talk about Native Americans and Theophrastus. There's also records of studying garden plantings by our first and third presidents, George Washington and Thomas Jefferson. George Washington reportedly wrote in his journal how he transplanted redbuds from nearby woods into his gardens at Mount Vernon in Virginia. In 1781, Thomas Jefferson listed redbud as in his only published book, Notes on the State of Virginia, as a native esculent tree. Esculent, I had to look up, is defined as a thing, especially a vegetable, which is fit to be eaten. And you can eat the flowers of Cercis. He intended Cercis to be a part of his shrubbery scheme for the western slope of Monticello and in the clumps of trees planted in the angles of his house. Monticello has, I saw like 80 Cercis around it, so it's quite popular there as well. Native Americans, of course, were familiar with Cercis and used it in looking, these are some incredible images of basketry. So it has ethnobotanic importance. It's highly valued by California basket weavers for the young wine red twigs, Plus the color in the bark can be used as fiber dye. These are images of some incredible Native American baskets, both made by Lucy Tells of Pomahas, meaning meadow, in her native Paiute language. Lucy was born as a Mono Lake Paiute and later lived and crafted in southern Sierra, Yosemite Miwok area. On the left is a beautiful basket which could be used to carry water. This basket is made of a willow foundation with a tan-colored sumac, Rus trilobata, coiled on top of that. The decorative designs are made of redbud and bracken fern root. This basket is in the collection of the Smithsonian's National Museum of American Indian. Excuse me 1 second. Where I live now in Nevada City is home to the indigenous Nisenan tribe. I hope in the future to learn firsthand from these tribal members about their cultivation knowledge and uses of redbud for food and basketry, but I wasn't able to yet. Kat Anderson, pictured here, has done an impressive amount of research, including many years of tribal visits and interviews, to understand and document how Native Americans tended or gardened the earth. Native peoples had a very, excuse me, I'm getting over a cold, had a different and active relationship with the land, which included regular burning. In this article, which is highlighted here, California Indian Horticulture Management Use of Redbud, she wrote that at least 20 different tribes utilize the branches and ground stems of redbud, spanning 22 California counties. This is her book. Incredible. I've seen her speak and really appreciated it. So the first written record in Western history is by Theophrastus. He's an ancient Greek philosopher and scientist. Around 3050 BC, Theophrastus began a 10-volume set of botanical treatises translated as Enquiry into Plants. These books constitute the most important contribution to the botanical science during antiquity and the Middle Ages. These books include an encyclopedia of the plant kingdom, which in a draft taxonomy is sketched together with the basic classification of plant elements. The work served as a reference point in botany for many years. In these books, Theophrastus named and described the redbud plant, which in Greek was called Circis. So Cercis is derived from the Greek Circis, which means a weaver's shuttle. And it's thought that for Theophrastus, the woody flattened fruit reminded him of a weaver's shuttle, and that's how it got its name. With that, let's look at some modern research. So here's species publications and taxonomic study. I would think most of you have heard Carl Linnaeus, and he published 2 species in 1753 in his seminal work, Species Plantarum. And then it gets a little bit more complicated from 1912 on. So I will talk a little bit more about all of these authors and what questions remained that drew me into studying this plant. Cercis is named in the classic and seminal work, Species Plantarum, published in 1753 by Carl Linnaeus. And in this work, Linnaeus established the binomial nomenclature system, which remains in use today with the genus and species. Before that, apparently there were very complicated plant descriptions that were pretty hard to list and organize, and kind of like common names, there can be a lot of overlap. In Species Plantarum, Linnaeus published the genus name Cercis and names for 2 species, Cercis siliquastrum and Cercis canadensis. Cercis siliquastrum is the Mediterranean species. It's sometimes referred to as the Judas tree, from which Judas Iscariot is reputed to have hung himself. Linnaeus gave this species the specific epithet of siliquastrum, which is derived from the Latin word siliqua, meaning pod. Linnaeus also included and named our eastern redbud, Cercis canadensis, which you see second here. He did this based on material that was collected in Virginia and then sent to Sweden, where he examined it. Finally, about 100 years later, Western science formally recognized our local redbud as Cercis occidentalis. And this publication marks the second North American species to be recognized. Cercis occidentalis was formally published in a treatment written by Asa Gray, reviewing a collection of plants made by a field biologist, Frank Lindenheimer, during the years 1845 to 1848. During this period, botanical science was becoming established in the academic towns in the eastern US, as opposed to Britain and Europe. So that was a big change. And collections could be made throughout the continent and then sent, in this case, to Asia Gray at Harvard for identification and nomenclatural documentation. There was always Latin included in the species publication, which you can see at the top of this. This was published in the Boston Journal of Natural History. Now that we have two species named in North America, I want to take a better look to see what kind of variation occurs and what questions remained unanswered. So looking at these two leaves, they do look pretty obviously different. And we would say that this one is dry adapted and this one is wet adapted. This leaf shape is rounded and could be called reniform or kidney bean shaped. Sometimes they indent in a little bit more. And this one is more heart-shaped or cordate. And it has this acuminate apex. And then the sinus is different. This one's got a more broad sinus and this one's got a more V-shaped sinus. These leaves also are thicker in the Western redbud. The leaf is thick and leathery. The surface is dull or sometimes with a light waxy coating. So this apex is thought to form what is called a drip tip and help in wet conditions to let water just trickle right off the leaf rather than accumulating, which can be good for it. Plants in between these two locations cannot always be so clearly distinguished. This is a variety Taxonomic, which has kind of an intermediate shape and the undulate margins. And they can be quite shiny. And then there's others, variety Mexicana, that has very fuzzy, compact fuzzy leaves at times. The branchlets also are interesting. You can see some are quite fuzzy, in botany call it pubescent, and others are glabrous without any trichomes or hairs. This is another morphological difference that's been used to separate taxa. The vestiture is different, meaning clothing. Found covering the branchlets or New Year's growth is different. So you can see clear differences with a light microscope. Now I'll spend a few minutes to summarize the outstanding taxonomic problems that I was working to resolve in my research. And to remind myself, the branch of taxonomy is the branch of science concerned with classification, especially of organisms. So there were a number of conflicting taxonomic treatments. So I'm going to mention 5 of the most recent taxonomic treatments on Cercis. Starting with E.L. Greene, who was a botanist that did work at California Academy of Sciences, where I did my work, and he also taught at UC Berkeley. He made a number of collections and named 7 new species of Cercis in one paper from 1912. So in 1912, with his work, we added 7 species. So in North America, we'd have 9, maybe 10 species. He did not have any collections from Mexico. Later in 1942, Milton Hopkins published a taxonomic review of the group, which I will discuss in more detail in the following slides. This is what's still currently used. Milton Hopkins with two subspecies. In 1975, Duane Isley conducted another assessment of the genus. It was preliminary, but he delimited 6 informal groups. So he kind of saw that they're all related, but that there were differences in 6 different groups. Next, in 1989, while writing a Cercis treatment for Intermountain Flora, Rupert Barnby concluded that the variation seen did not warrant taxonomic separation. So he said it's just one species. Finally, my advisor and some students working with him looked again, and he included specimens from Mexico. And what he found is that there are two distinct species based on morphology. But then he brought me into the lab and he's like, there's still a lot of questions that are outstanding. Here's the journal article for Hopkins treatment. It published in the journal Rhodora, the New England Botanical Club. In this review, Hopkins assumed some of the earlier designated species that Lee Greene had done based on trivial characters, and he reduced the rank of some taxa as he found no justification for their separation as species. So he's got the two species, and then he reduced Taxensis and Mexicana to varieties. This has become the fundamental study that is still relied on by most researchers and field biologists. This treatment resulted in the recognition of only the two species. It attempted to treat the genus in all of North America, but in fact underrepresented Mexico due to the lack of collections and the reliance on poor distribution maps. This shows the 4 different groups that Hopkins recognized. We've got the 2 species, Cercis canadensis, throughout the East and and Texas, and the plains here. And then our Western redbud, Cercis occidentalis, which extends here into Utah and Arizona. He also saw that there was variation in these 2 groups, Mexicana and Taxensis, which have different symbols here. And then Duane Isley looked at this again, like I had an overview slide, and he delimited 6 informal groups. Overall, he concluded that the variation in the genus is clinal or continuously changing over space. He did not attempt to revise Hopkins' taxonomy. Instead, he published some tentative results and recommended further study for the group. His review was specific to the US, although he referred to and recognized the need to do further research in Mexico. And we felt that that was essential to understand the evolution in the group, is the variation seen in Mexico. So Isley did recognize that this Colorado Plateau group is a little bit different, and he saw variation in San Diego, which I did not find. 1989, Rupert Barnaby. He looked morphologically at the group and found that the variation seen could be accommodated by one species with strong regional expressions. He also did not include Mexico. So there's the one species for North America. Then in 2009, to better understand the genus, my advisor set the stage for my research project. He did do a pretty comprehensive study of Mexico and looked at the leaf shape differences. But he felt like we needed to combine this data set with new data from the Western US to get a good sense of what's going on. So that leads me up to my research. 1 second here. Right, hopefully that will ease the throat. With that background, I'll spend some time discussing my and my colleagues' research in this group. I want to emphasize that all aspects were very collaborative. Throughout the project, I worked with my advisor, the molecular lab manager, and was able to work with and incorporate the data collected by interns and a volunteer that had also worked on this project. So to address the outstanding questions, I did what can be separated into a 3-part study. I conducted a detailed morphometric study using air-bearing specimens from across North America. I used the morphological data set and GIS to test the ecological value of leaf traits. As you recall from the leaf slide, some leaves show dry adaptations and some wet adaptations. To test the adaptive value of leaf shape, I conducted modeling with environmental variables. Then finally, with my colleagues at California Academy of Sciences, I constructed improved molecular phylogeny to show the evolutionary relationships for the genus in North America. To begin this project, once I had some theoretical understanding, I needed to start by making a number of collection trips throughout the lower Montane, California, and to the 4 western states where western redbud naturally occurs. This was in summer 2011 and continued into summer 2012. 4 large trips, one to throughout North America, sorry, Northern California, one throughout Southern California, and then another one flying into Vegas and collecting these plants here. This was to obtain fresh leaf material for DNA analysis and to gather herbarium collections from under-collected regions. I will give praise to my invaluable field assistants again at the end, Angus McGrath, Alan Kastner, and Diana Gamba Moreno, who were also great fun and kept this project going. Here's me in 2012 beside a bushy western redbud in Northern California. You can see it's nice and tall. This is preparing a branch as a collection specimen. This is the botanical field press that I borrowed from Cal Academy for all these trips. So once I had a bunch of fresh material, I worked on the morphometric analysis. So I will go into more detail about this. The word morphometric means a quantitative analysis of form or size and shape. The largest component of this research was conducting this continent-wide morphometric analysis of the genus. I did this with the herbarium specimens, which you can see pictured here. As many of you know, herbarium specimens are pressed and dried plant cuttings with detailed collection information. They're used to document and analyze global biological diversity. At the California Academy of Sciences, there is an excellent collection of over 2 million specimens of all kinds of vascular plants. Plus, for this study, we used loans from 10 other herbaria. So with these specimens, first we had to georeference all of them. And here's the collection information with the date and the biologist that collected the plants. But it's got a regular description of the location, 3.2 miles west of US Route 20, north side of the French. I can't quite read that size, but so we had to translate this written narrative of where the plant was into latitude and longitude. So that took quite a while to get these 11,000 collections georeferenced so that we could plot them and do analysis that way. Fortunately, I had the help of other people to do this North American data set with summer systematics interns. Anna Schiller, she did Texas and Mexico. Lila Leatherman did the Eastern United States. And we had Sue Vocal with the Western United States and doing leaf area. I was trying to lose my voice here. So we measured structures of the leaf, the fruit, and the flower. And we did 2 leaves and 2 fruits and flowers per specimen. We did the length and the width of the leaf, the length, width, and wing width of the fruit, and then the flower for each specimen with digital calipers. Here's the flower length, which is kind of a rough measurement, but with such a large data set, it does show trends. And then here you can see that we were measuring the wing width, which does vary across across different species. To standardize, we measured the 2 largest leaves, not just anything there, because there could be a lot of variation within one collection. We also looked at the leaf shape quantitatively. The leaf apex and sinus shape were scored against reference shape shape categories obtained from Peter Fritsch's Texas and Mexico study. As with the other size characters, to standardize, we measured the two largest leaves. So we're looking at the apex and giving it a number to see if it was a cuminate, more of a drip tip or a rounded leaf. And then the sinus shape as well to see if it was very deep or more shallow. We also used this Leica compound light microscope to look at micromorphology of specimens. Recall that some plants are very fuzzy or pubescent. The small branches, undersides of leaves, and pods show obvious variation in this character. To quantify the variation for analysis, we outlined a 2.5 millimeter square area on the underside of the leaf. Within that area, we counted and recorded the number of trichomes present. And you can see here the fruit surface does vary. This one, if you blow it up, shows clear, tiny hairs or trichomes. This has none. And here we have a leaf surface, which is glabrous versus pubescent. There's the 2.5 millimeter square. Finally, leaf area was captured through imaging and analysis. With the leaf area results, our data set was complete, so we could start performing a variety of analyses. And this is plotting all of the collections and the variation that we saw before we started to analyze the trends. To assess the variation in the genus, we constructed a character matrix based primarily on the taxonomic treatment of Isley. The specimens were separated into 6 groups, plus unknown in gray. So you can see a little bit of unknown here. Those were just plants with just flowers, so we couldn't analyze them as much detail if there were just flowers present. These are our 2 Western groups, which we called Colorado Plateau and Cercis occidentalis, OCC and CP. This is Can for canadensis. This is Can 2, which was also wet adapted. But this set of characters was only found in this Sierra Madre Oriental group down here. And then we had Tex and Mex, which had somewhat different characters that are overlapping. So this point, Colorado Plateau from the Grand Canyon and smaller canyons found in Nevada, Arizona, and Utah. This group will become more significant later. So let's think of that one. Once we had all of that in a data sheet, a database, the morphological data was analyzed in a number of ways. We did univariate statistics for leaves and flowers, which were visualized using these box and whisker plots. The first plot shows the character of leaf length on the y-axis. So here's leaf length. And these are our groups moving from west to east across North America on the x-axis. The central 50% of values is within the box. And the whiskers encompass the range recorded for each group. So occidentalis is 50% here, and that has this range. You can see Mexicana has a very large range, as does Texensis and canadensis. So very long leaves and very short leaves. And then occidentalis has just very short, small leaves. So you can see there's a lot of variation within the groups and the regions. The second plot shows the character of flower length between the 6 groups. This character is interesting in that 2 geographically separated groups can have substantially longer flowers. The Colorado Plateau group had the longest flowers, as well as Can 2 from southern Mexico. We also analyzed the data using multivariate methods. Principal components analysis is used to find the underlying patterns in a data set. We use this to examine the variation to find out if the characters we observed could be used to separate the taxa. The scatter plot was constructed from leaf, fruit, and pubescence data. Flower size was not used, and color-coded to show the geographic groupings. We can see that 4 of the 6 groups occupy a nearly continuous band and overlap at the bottom of the graph. In contrast, these 2 groups are separated vertically on the y-axis. So this is Mexicana and Can 2. But this is actually just due to branchlet pubescence. So these have fuzzy twigs. Otherwise, they would all cluster together. And we didn't feel that the fuzzy twigs was a reliable taxonomic character. So initial results from these analyses suggest that the range of morphological variation in Cercis is clinal. The results showed that leaf shape varied continuously throughout the region, showing ecological adaptation. We felt that the leaves were correlated to climate, which I'm going to get into a little bit more next. Like you saw in the box and whisker plots, the flowers are larger in the groups Colorado Plateau and Can2, and pubescence can be used to separate out two of the groups. Next, we did an adaptive trait analysis. A second component was looking at how the leaves are adapted to climate. Based on the variation observed, we hypothesized that there was an association with environmental conditions, specifically temperature and precipitation. We looked at 3 leaf traits using GIS. The question is, are leaf traits adapted to the climate conditions? And we tested leaf area, the apex shape, and the sinus shape. 6 layers of climate data were obtained from the online database WorldClim. These layers are compiled from hundreds of global weather stations and interpolated into high-resolution continuous raster files. 2 baseline layers were used for temperature and precipitation, and then others were added that more accurately account for seasonal stress due to summer drought conditions. This is leaf apex shape plotted here, which we're hypothesizing is adapted to climate with the drip tip. So you can see the more pointed leaf tips are clustering here in the darker green and throughout here, which can be quite music. And then the rounded tips are all found here in drier areas, which is not surprising, but it's interesting to look at it through data. We used GIS to extract the climate data at each specimen collection site. And we analyzed the data using multiple stepwise regression. So a preliminary study of this, what we found conducting these 2 analyses is that morphological variation is continuous and shows clinal change over geographic space. No single character or set of characters can be used to clearly separate the taxa. The variation occurs to be more strongly correlated with differences in latitude and longitude. However, subtrends were found. The leaf size and shape varied continuously throughout the region and showed correlation to climate. Like I've said, the flowers are larger in the 2 groups. We suspect that the flower wing and lateral petal length could be informative in the Colorado Plateau in the Can2 group. Pubescence can be used to separate groups, but the distinction of this trait is scattered in geographic space. It does not seem to be confined to closely related plants. So essentially, we found continuous character variation throughout North America. This has also been proposed in previous studies. So should there just be one species in North America, which supports Barnaby's species concept? And Isley also hinted at that. However, there's another way to examine the variation seen in the genus, which is what we did next, a molecular phylogenetic analysis. The last major component of the research was to create an improved molecular phylogenetic analysis. Phylogenetics comes from 2 Greek words, phylon, meaning tribe, clan, or race, and geneticos, I'm sure I'm not saying that correctly, but meaning origin, source, and birth. So we're looking at tribes and what their origins are. We were seeking to better understand the evolutionary history and relationships within North America's Cercis. These relationships are discovered through inference methods that evaluate heritable traits such as DNA sequences. Previous phylogenetic trees have been created for the genus, but were limited due to so low sample sizes and because there were no collections from some important regions. In our tree, this was improved by the addition of 21 new wild collections from the Western US that I got in those big expeditions in 2011 and 2012. So with these, we employed DNA sequences from 4 genic regions, the nuclear ITS gene and 3 chloroplast intergenic spacer regions that have worked well in previous studies. We would mix minute volumes of DNA and reagents in strip tubes such as these. In the interest of time, I'm going to skip the detailed methods and go right into our results. So next I'll present our phylogenetic trees, basically a diagrammatic hypothesis about the history of the evolutionary relationships within the group. So here is the overall Northern Hemisphere phylogeny that includes the plants from Asia and the Mediterranean. This tree was created using the programs BEAST and Mr. Bayes. It uses Bayesian inference methods to estimate divergence times between clades. The divergence dates are plotted in black. So here we have million years of time. This is at 50. This, that's our first outgroup. This is all of our Asian species. And that divergence date is estimated to be 27 million years ago. And the clade credibility support values are plotted in red. So this separation is strongly supported. Most of these have really strong support values. I think that's one of the weaker ones, but it's still quite good considering what we were looking at and the data we had to work with. We used 2 fossils to calibrate a molecular clock by scaling the length of the branches to a geologic timeline. So here's one fossil, which is used to calibrate the molecular clock. This is a legume fruit with a wing, which is a more recent form of Cercis. This was estimated at 36 million years ago. Then fossil 2 is a Xeric with a minimum divergence time of 11.5 million years ago. So the nodes could be older but not younger based on these fossils. And then I'm going to highlight. So this is Eastern Eurasia. This is China, basically. All of the species there. And then we have Western Eurasia with Cercis griffithii and Siliquastrum in the Mediterranean. And then we have all of North America, which, oops, oh, it's not, okay, which is clustered at the top. The next phylogeny is going to show just North America.
I'm gonna pee myself.
So here's North American phylogeny, focusing in on this. And I'm going to highlight these. So here's California, all of the recent collections, and they strongly group together. There's little bits of, I think, insignificant variation in here. I don't think any of these subgroups meant anything, showed anything interesting. And then we have Eastern North America. That's all the typical Cercis canadensis, including some from Mexico and Texas, and says including some of the dry adapted forms, but they all clump together. And then we have the Colorado Plateau. So this is a really interesting result because if you go and look at these plants in the Colorado Plateau and the Grand Canyon, they look very much like our Cercis occidentalis, our Western redbud. And that's what they've been considered up to this research. And we haven't quite overturned the paradigm yet, but that's what we're going towards. And this shows a separation of 18 million years. So a closer relationship exists with the Eastern redbud between Cercis occidentalis and the Eastern redbud than this. There's no kinship here. So convergent evolution is occurring according to these results. So a results summary, morphological and adaptive trait study. I found that the morphological change is continuous in North America, and the morphology seems to be closely tracking environmental parameters. Based on morphology, there's no clear justification for multiple species. So perhaps we should treat Cercis as one species in North America. When we add the molecular phylogenetic study, we're we're left with conflicting results. The molecular data indicate that there are 3 clades or presumptive species in North America. Colorado Plateau clade diverged from the California clade in the Miocene about 18.1 million years ago. So this makes you wonder, is there cryptic species occurring? And how common is this? So here's another summary slide. Cryptic speciation occurring. Is there just one species only? That was supported by the morphological data. Are there 2 species? That's the current circumscription that most botanists use based on Hopkins. None of our work confirmed that or supported that. Or are there 3 species? The Western redbud, the other Cercis, we're calling it Cercis orbiculata from the Colorado Plateau, or and plus the Eastern redbud, which includes all of those others which are currently supported, which are currently under Cercis canadensis. That was supported by the molecular data. So cryptic species was one of the interesting results here. It's called the biodiversity wild card. 2 or more distinct species that are erroneously classified as a single species. This review from Trends in Ecology and Evolution documents the increased recognition and referencing of cryptic species since the mid-1970s. When PCR methods came into widespread usage. This review argues for the importance of scientific and taxonomic recognition of cryptic species. As such, species recognition is fundamental to implement conservation and best management practices. Another more recent paper in a botanical journal, Taxon, argues that cryptic taxa should have names as they are on a separate evolutionary trajectories and should not be conflated with molecularly or cytologically dissimilar groups. And here is the paper that we published about all of this. This was done in the American Journal of Botany in September 2018, years after I graduated, but we're very happy to get it out there. My advisor is the first author, and I'm the second author with a number of the other people that contributed to this. So you can download this online. It adds a lot to what I've said tonight, but we're very happy to have it out there. Applications of this research, which I hinted at a little bit earlier. This and similar studies can be applied to broader scientific questions. For example, testing models of historical biogeography. How did Earth end up with the current distribution of plants? I spoke about this a little bit. And related to this is estimating paleoclimates and predicting the effects of climate change. A strong correlation that was found to climatic conditions can be used to model and better understand both ancient and future climates. We do have some good fossil collections dating back to the Eocene from beds in North America, which can be used to infer early climates and see changes in climate over time. The fossils are from beds in Oregon, Washington, and the Florissant in Colorado. So with that, I want to present some acknowledgements. I want to reiterate that this has been a very collaborative project, and I had a lot of excellent support. I want to thank my advisor, Dr. Peter Fritsch. He's now at BRIT in Texas. My lab manager, Bonnie Cruz at Cal Academy, and my committee members from San Francisco State University, Drs. Jerry Davis, Tom Parker, and Bob Patterson, as well as the other collaborators from the Botany Department and the Center for Comparative Genomics Lab. And I want to thank my invaluable field assistants, of course, Diana Lucia Gamba, here on the left, in a field of flowers. After our work together, she completed a doctorate degree at the Missouri Botanical Garden and is now working on a postdoctorate. Alan Kastner, who may be joining our meeting tonight, is a good friend and was an enthusiastic and intrepid field assistant. He's now a forestry field preparation specialist with the California Tahoe Conservancy. And Angus McGrath, who I call Gus, in the middle here, was an incredible and moral support and field assistant throughout the project. And for the last 6 years, my amazing husband is shown here in the Grand Canyon. I also want to express my appreciation to CNPS, Redbud Chapter President Shane Hannafy, Chrissy Freeman, and Jean Wilson for inviting me to speak tonight and for hosting many informative and wonderful native plant programs. I'm happy to take your questions and hear your feedback on this study. And here's one more slide. Feel free to reach out to me. I've been working with CardNote, which is now required and merged into Stantec. So you can reach me at camille.noel@stantec.com, or my profile's on LinkedIn. You can message me there. So thank you.
Thank you so much, Camille. You know, when we were doing the preview of this, kind of our little practice sessions, I don't know, a month or so ago, you know, I was just amazed at the depth of research that goes into advancing botanical knowledge. And I think that in some way, when I've forgotten all of the details, I'll still remember that. And I hope that that's true for other people as well. So here are just a couple of questions. First of all, if anybody has any questions, please do go right ahead and post them. And we will pass these on to Camille. I'll be sure to speak them out loud. So you alluded to something at the end that I think, as you said, you know, it does have implications for what we may be able to learn or understand about our world. And that is, what do you think there is about how Cercis, for instance, traveled from one place to another and then evolved as it went from one place to another that could help us understand what might happen with other species in response to climate change?
Yeah.
I realize that the pace of millions of years ago and climate change are not at all the same, unfortunately.
Well, yeah, definitely there was very long time periods that this evolved over. So in the evolution of this plant, there's two hypotheses. One were land bridges, that there's a North Atlantic land bridge and a Beringian land bridge, that the plant could have like had intermediate plants on those land bridges. And then as they went into the ocean, into the North Atlantic, those intermediate plants are gone. And the same thing could happen in North America, that there was more continuous bands at different climatic periods. And then as the climate and the habitat changes, we don't see those currently. So I think it's a coevolution. The plant co-evolves with the climate with time. And this study lets us think about it in different ways. But I'm not sure at the rapid pace of current climate change, if Cercis has all the answers.
Yeah, absolutely. And what are some of the hypotheses about the spaces in between? You know, there's a Cercis, there's Cercis in this space, and then for 1,000 miles there's no Cercis, and then there's Cercis, and then for 700 miles there's no Cercis. Could you help us understand what you all understand about that?
Yeah, I don't know exactly. I mean, it does need a good rain, you know, and good soils. And so I think it's thriving where it's best adapted. It doesn't go into Oregon at all. It doesn't really extend past where I've shown it, doesn't go into Canada. So, you know, it does have restrictions in what it's adapted to. And it may have died out due to harsh conditions.
One of the participants asked a related question, which is, what's the highest elevation at which you were able to observe Cercis in your research?
Well, I don't remember specifically, but I think about 4,000 feet.
Okay.
Okay. I mean, I've yeah, I could look at the data set and see if we had anything higher, but I don't think it gets too much higher than that.
Absolutely. Okay, well, and here we are. Oh, and somebody asks, while we're still in that part of the world, where is the Vegas population?
In Red Rocks. So if you fly into Vegas and go to Red Rocks, which is just to the west of Vegas, I mean, it's only half an hour or something. It wasn't a big population, but there was a few plants there. It's a beautiful area in a state park there.
That sounds,
yeah.
oh, it was like finally a reason to go to Vegas.
I know, exactly.
For some of us, you know.
Yeah, there's not many in the Eastern Sierras, but there was a couple there. Yeah.
Okay, that's fascinating. Absolutely. And what have you studied enough about the Cercis occidentalis that's here in Northern California, where we might be personally interested in, to give us a sense of to what extent there are ecotype variances or variations that should inform our own decisions about where we get redbuds from and where we collect redbud seed. Not that we don't have a billion, you know, but.
I didn't find anything really, I mean, I would see different types of plants at times, and it wasn't something that we had enough time and resources to kind of see if there's ecotypes. It's definitely something that occurred to me at times, like, why is this just, sometimes I get to washes and they would just be dense with like young redbud, old redbud, you know, all kinds of redbud. And other times they were kind of few and far between. So I don't know exactly what the variation is, but we didn't find anything that was measurable with the morphological analysis.
Yes. Yes. Okay. Okay.
What's the lifespan?
Sure, go for it. And how does that vary? Yes.
I believe around 15 to maybe 30 years, but not a long-lived plant. 15 to 20 might be its peak health.
And oh no, that was going to be a question about you know, the use of them in the landscape. Oh yeah, okay, sorry. Oh, could you talk about Cercis in terms of its traits as a fire-adapted plant? Is that anything that you know anything about? It's kind of a cross between what do you know as a scientist and what are we interested in knowing?
Well, I know that Native Americans burnt the land. So, and they would coppice the plant. So I think as long as the root stock is protected and that there's still some root, it'll regenerate. But that's a good question. I should learn more about that because I know that the hills were burnt regularly and the Cercis is abundant, I think, without any hard burns.
Right. And the question that we were going to ask was about coppicing. So I decided that would pass. Nature doesn't exactly do that, but it does do the fire. George Chang asks, have you seen the white flowers of the Western redbud?
I think that's more of a cultivar. I've never seen white flowers. I know that there's like really deep purple, like red and stuff, but I think those are all cultivars.
Okay. Okay. Let's see, what else is there? What else would you like to study and learn and have others study and you be able to learn about Cercis?
I would actually like to learn more about pollination. I haven't, I was looking for more, looking for studies on pollination, and I don't know what I even use that it's pollinated by bees. I think there's a few kind of anecdotal things, but I've not seen any good study and documentation of what pollinators. And I didn't see many bees on the plant. So, and also the fruit stays on the plant sometimes for 2 years, which I thought was kind of odd that it wouldn't just fall and so yeah, how it like reseeds and the pollination is something I'd like to know more about.
I can see that you are far from the only person that we have other comments about this. So another question about, you had said that redbud are not nitrogen fixing. Do they fix nitrogen without nodules, or is it that they're not nitrogen fixing at all? What is their relationship to this? Do you know?
That's a good question. I mean, I just don't think there's been any nodules found, and there's no reports of people studying or reporting on this. So not all fix nitrogen.
Good.
So they must just be in nitrogen-rich soils that have enough nitrogen for them to grow.
That's right. So they're not dependent on what they pull out. There has to be some there. Okay. Yeah, they're not generating. Any other question? Let's see here. We do want to remind people that yes, this is being taped, and it will be up on our Redbud YouTube channel, and not very long. Thank you so much to Shane, who is always taking care of that for us. And would you say that, you talked about the different leaf shapes, and the pointier ones are where there's more water, and the rounder ones are where there's not water because the water doesn't have to drip off the leaf. Have you seen that in our own Northern California redbuds? Now you're here, you see them all the time.
So yeah, they do have the more rounded, the apex is rounded, right? So that would, I mean, there's just, the water would kind of flow all over, but in a really wet forest, it channels it to the tip. So that's what I saw, but that's what I've seen in the wild and with the herbarium specimens. I'm not sure if I'm answering the question correctly, though.
Not quite sure. We'll see what happens. Somebody who saw your images of the fossils from Cercis was the first example of the pod or the leaf. They apologize if they missed that you said it. Yes, of course.
Yeah, there's both the pod, the long pod with the wing, and the wing. I would need to check in with my advisor a little bit about this, but I think that wing is a later adaptation, and I don't know when in the genus that was first seen, but that helped us to put that on the phylogeny when that wing was first seen. And then the other one was the dry adapted leaf form, which is the more recently evolved type of leaf form in the genus.
For those of us who are not familiar with the evolution of the tree species, the shrub species that we have now, how does Cercis line up in terms of being early or late in that evolutionary race, so to speak, in that process,
So the Western is a more recently evolved form. It seems that the group originated in China around 34 million years ago, and then went west. So then it went to Central Eurasia. There's another species in Afghanistan. And then, so yeah, the whole Northern Hemisphere phylogeny shows the Chinese species clustered together, and then the Afghan redbud, and then the Mediterranean redbud, and then more recent divergence shows the North American species. And of course, they used to think it all originated in North America around Harvard or something, but but we, with our research, think it could have originated more in Mexico and then moved northward from that somehow it got into Mexico and then kind of grew into North America, into the three different groups.
Well, I think that does it for our questions, unless somebody posts one in the next 15 seconds. It's just been great to have you here and help us have a deeper understanding and appreciation for our redbuds. And when we see the eastern redbud in people's landscapes, then I'm sure we'll be taking a closer look at the leaf shape and the size of the leaves and so on and so forth. So thank you so very much, Camille. And we really appreciate everybody who came this evening. And thank you so much to Jean, and thank you so much to Shane. And we'll see you at another Redbud event.
Thank you, Chrissy. This has been a lot of fun. I really appreciate it.
Oh, great. Okay. Thank you all.
Bye. Bye, Kate. Great to have you here.